I'd now like to call the participants of our panel, Dr. Alice, Professor Evanguedes Kalapothakis, and Dr. Milton Barbosa. And I would like to thank our guests and the audience for participating in our event. Thank you, Gabriela. It's a pleasure to be here at this event. Thank you for joining us, Professor. Professor Newton. Hello, Gabriela. Thank you. It's a pleasure to be here as well.
Dr. Alice Valentini, can you hear us? I think your audio is off. Would you mind switching it on?
Your microphone is...
sorry. Hello, everybody, and thank you to let me be part of this event.
Muito bem-vindos. Vou deixar agora...
Welcome, everyone. I am now going to hand over the panel to Milton Barbosa, and he's also going to ask the questions. So if you want to ask questions, just send them through the Q&A tab, and have a great panel. Good morning once again. We are here live now. It's a pleasure to be here with you. Thank you once again from CBEIH for inviting us. I loved the presentations. I received lots of compliments. Many people enjoyed it as well. An interesting thing about these new tools in the environment. I have lots of questions here from the audience, and I have some of my questions as well, and I'm going to start by putting a question from our friend Hayan. It's a question for Dr. Alice. When you make an analysis of eDNA, many samples come back as not classified because of the consequences of previous things that we have in the database. These sequences that are not classified, could they be new species?
Maybe new species, but can be also new haplotype for one species that diverged during the time, and after in some population the DNA is different from other populations. So even if the species is present in the reference database, you can miss it because, for example, the species that was sequenced in France, but not in Brazil, so you can miss the species for this reason. But also can be a new species or a sister species. So they thought it was one species because of morphology, but the genetic point of view, they may be two different species.
Inclusive uma espécie críptica, imagino.
And also a critical one, right?
Yes. Yes, of course.
Acho que está respondido, né, gente?
I think that's the answer. Thank you very much. Thank you, Alice.
You're welcome.
I have a question about this technical topic for Professor Evanguedes and for Dr. Alice. What are the challenges that we have today to be able to implement these technologies in the public policies of different countries? What are the challenges that you face? And if you can talk a little bit about that, about the public policies that have to do with environmental monitoring. What is the prognosis of using these tools in a widespread manner to monitor the environment in all the countries around the planet?
Dr. Alice, if you want to start.
Okay, yeah. From European point of view, there was a cost action is a kind of group. It's a project that brings all the researchers around Europe together to the main goal of this project was to evaluate the eDNA techniques for water framework directive for fish biodiversities and the other group for detecting this. So this was done at the European level, and now environmental DNA in Europe is considered as a manner to do in parallel than traditional methods. Also in France, it can be used now for environmental DNA, environmental assessments. So there are not so many challenges for species detection for this moment. Then the technique should be validated before using by pilot study. But once it's validated, and that can be used by people monitoring, using by officers that do environmental impact studies, and also stakeholders. And as the example for the Great Crested Newt in UK is now detected only using environmental DNA around all the England. But there are some challenge. For example, the quantification of the DNA is still on debate. On the quantity of DNA that we found is proportional to the biomass of the species in the environment. So the study now are focusing more on this second part of the question about it, because environmental DNA now demonstrated that is suitable for species detection, even rare species, but the quantification part is another part to be investigated right now. I don't know if you want to add something else, Dr. Evanguedes.
Yes, sim.
Yes. I think it's important to consider that the environment of a tropical country is different from a country in Europe. So this brings some changes, some practical changes in the process. So I believe that to be able to get acceptance of this technique by the scientific community, by society, by the companies, we have to work, for example, on a pilot project. And this pilot project has to bring knowledge, practical knowledge, compared to the traditional methodologies, as seen in several of the publications shown by Dr. Alisi. And this will generate reliability. And with this pilot project at this moment, we could actually have another encounter with the strong presence of people connected to the release of these projects, and also politicians. That's where they would see several aspects, several positive aspects of the methodology. The sensitivity of the methodology, and also the costs that are generated, as shown by Dr. Alice. They can also be much lower compared to the traditional methodologies, and also make it clear to avoid, at least in the beginning, that the eDNA methodology works together side by side with other traditional methodologies. So one does not exclude the other, they complement each other. So many people believe that in the future, we're going to have only eDNA monitoring, right? So it's not really true, because they are complementary, and we're not going to have one being excluded, right, out of the story. They're going to move together, right? Yeah, there's always going to be a new demand, always. And What is the demand? We don't know what it is. It could be, for example, coronavirus or any other parasite that comes up. So what is going to appear, what the new demand is going to be, it's going to come up definitely. And at this point, it's going to be very important to have several professionals working together to determine the best way of using technology, eDNA technology, in a practical and functional manner. I'm going to take advantage of this topic. Would you like to add something, Dr. Alice?
Yes, just very quickly, eDNA can be used as a fast screening of what's in the nature, and after you can go back with traditional method to find, to refine the question that you are asking. For example, some question can never be answered by eDNA. For example, if you are interested on the age of the individual that are present in the environment, this kind of information the eDNA cannot be done. So you can just screen the environment to find where the species is present, and after go back only in the place where the species is present to do some traditional trait-based analysis like the length of the fish or the age of the community and things like this.
Sim.
Yes. Erica Barum is asking, and I think it fits into this discussion. It's about the plants. So what is the feasibility of the collection of eDNA for plants? Can we actually make collections of plants using eDNA? Is that possible? It's a question for both. Yes, I'm going to give you an example of what we're starting to do when we talk about forensic analysis, human forensic analysis. So nowadays, we already have an expectation of collecting DNA from the air in crime scenes. So this means that when a criminal goes in, he releases his DNA, his cells, and when he releases very small particles, these particles, they float in the air for a while. And there are already groups working on air filters, and they collect volumes of air that concentrate the particles in a filter in the same way as this is done in water. And from then on, they make an analysis. So yes, it is possible for this to be done. Even for plants, because I think that plants, they probably leave maybe less DNA fragments in the environment. You can think, for example, in pollen. That's just an example, right? And in the same way, the plants go through degradation. So I don't know the efficiency, right? Excluding pollen, which we know that is very abundant. Maybe Dr. Alice can tell us. Yeah, and resistant as well. I don't know if Dr. Alice has some information about that.
Yes. Plants' DNA can be extracted from environmental samples. So there are less study on water samples, but on soil sample, there were few studies using environmental DNA collected from soil samples to describe the plant biodiversity on French Alps and French Guiana too, and other places. That was quite similar to the botanist inventories done in parallel. And also it was possible to collect DNA from what is called environmental samplers. Is actually the animal that eat the plants, and so analysis the scats of the animals, you can find the plants, or even honey. For example, we during my PhD, I extracted the plant DNA from honey samples collected in the market, and it was possible to describe the plants found in the honey. So plants DNA can be extracted from an environmental sample, and there are less study on it, but it can be done.
Sim, imagino que em ambientes.
And I think that in more humid environments like tropical forests, it's even easier, right? Because, you know, compared to dry areas for plants, that this really depends, because we always have to consider the volume of DNA that is released and the stability of this DNA. And whenever there is greater humidity and a higher temperature, you favor the growth of microorganisms that release DNases. And it's not few, it's not just a few microorganisms that do that. So you, for example, can have a sample, a new sample, relatively new DNA sample that was completely degraded, right, in a few days. And we know that there are samples that are found with more than 1,000, 2,000, 3,000 years of age, and they still generate reliable outcomes in terms of sequencing. And that's, as we say, it looks like magic, right? In a way, for us to be able to identify things like that. We talked about plants, and we have a question over here from Hudson Paixão. And he's asking about examples of detection of birds with eDNA. A question for both of you again. Dr. Alice?
Actually, there are very few publications of birds, but they can be using what we did for French Guiana, for example. We use a universal primer for vertebrates. So we have we show only mammals species because the student was most interested on mammalian species, and because we have the comparison with the field only for mammals, but we found lots of species of birds. And so is it possible to detect birds? But there are, I don't know why people are less interested. And from my knowledge, there are less bird publication than fish publication.
Nós temos, junto com...
We have, together with IBAMA, Dr. Alice, that's an organ that monitors the environment in Brazil, inspects the environment. We have a project that is being developed together with the Brazilian Academy, Forensics Academy. And IBAMA developed markers for birds. And especially because of traffic, but also in terms of conservation. So one example with regard to a bird called bicudo that exists in a large quantity in the areas authorized by IBAMA, but they practically disappeared from nature. And with the development of these markers, it is already possible to work with feces or with feathers or any other thing to try and identify the presence of these birds. In the case of bicudo, this bird, this is being done, and we also have with tricaferro or with parrots and other birds. But usually they are forensic analysis. Now, with the forensic analysis development, and normally we use degraded, very low quality specimens, it is possible to apply the same study with DNA. And I think that birds have a characteristic of some populations have a very small population for many years, right? That's a characteristic of birds. So these tools, they help a lot in considering them. There are several questions here. I'm sort of trying to include all of them. There's one, a very interesting one here from Ryan, our colleague, for Professor Evanguedes. And the question is, because of the great quantity of DNA in a water sample, maybe it's not going to work because there could be some masking of the target. And what about the dilutions of the sample? Is this a problem for the target DNA? I'm not sure I understood the question. Could you please repeat it? Because of the large quantity of DNA in all water samples, PCR maybe is not going to work because the targets could be masked. Did you understand that? Yeah, well, the trend is for it to work. It works very well because, in fact, when you use a PCR, you increase the specificity because, first of all, you use external primers that already have a specificity, and then you use primers that are internal, and they have their specificity. So the technique in terms of specificity and sensitivity is excellent. Now, the great risk that we have that normally happens is that you increase the chances of a false positive because any small level of contamination during manipulation or from your lab, it's going to generate a positive outcome. And that's exactly what happens with qPCR techniques. Because of fluorescence, for example, a technique, any basal level of contaminants can be detected. There are ways of reducing contamination, of course, but this is the great problem, and it is the main reason for people to tend not to use nested PCR. Thank you. Okay, so I came...
Sorry, this is also the case why the environmental DNA labs should be constructed in a way that is more similar to ancient DNA labs, because your rare species DNA is the same level that your contamination. So special precautions should be taken when you using environmental DNA for rare species. Because you can use environmental DNA for diatoms, is still environmental DNA, but the quantity of diatoms DNA is so high that you will not see the contamination. But when you're using for rare species, the constraints are too much that you really need the special equipment. Sorry.
Yes. And talking about equipment, Roberta asked a question. She is thanking the speakers. She congratulates the speakers, and she asks a question to Dr. Alice about the collection of environmental DNA in streams. With the quantity of water in liters, it's filtered, and if there is special equipment for the filtering, or is that done manually?
No, actually we collect 30 liters of water per sample, and we collect two for stream. I'm talking. So we collect 2 samples per stream for the majority of the project, and we collect 30 liters per filter. That means 60 liters per stream. per site in the stream. So you can do it manually, but it will take very long time. So we use a peristaltic pump for pumping, and we have 2 or 3 times, but the easiest one is a kind of a drill that it was modified with a peristaltic pump in the front. So you can use it like normal drill that you use for your work at home and things like this. And you pump it for 30 minutes. And the collection flow is 1 liter per minute. So that allows you to collect 30 liters in 30 minutes.
Newton, eu poderia fazer uma pergunta?
Newton, can I ask Dr. Alice a question? Yes, sure. Go ahead. Dr. Alice, I saw in your presentation a picture of a boat, and I cannot imagine the size of this boat with the adapted filter at the back. So is this filter for collection? Is this a boat that has remote control, or am I wrong?
Yes, actually it's a pump with a boat shape, and the filter is this size, more or less, and the boat is less than 1 meter long. And so you cannot move for the moment the boat, but you can put it in the middle of the river from a bridge, for example, and there is a remote control to start the pumping and a remote control to stop the pumping, and after you can collect back your sample.
Ok, obrigado.
All right, thank you. That's very interesting. So we can imagine that we can make these collections in tropical forests with boats without a crew, right? In large distances. Yes, and with drones, also with drones. Yeah, to collect water and air, as you said. Yeah, that's right. Very interesting. And sorry, I forgot my question, but anyway, I have another question over here from Alan for Dr. Alice. He also congratulated both presentations, and he wanted to know a little bit more about the information analysis after NGS for coding. I think that's also Professor Evanguedes can also add on to this.
I didn't understand the question, sorry, can you repeat?
Sim, ele diz assim.
He says this: I would like to hear a little bit more about the bioinformatics analysis after NGS.
Okay. After that you collect your sequence after the sequencing, you have something like millions of sequence corresponding to your sample. So the way you need to analyze this, and so there is a huge part of the analysis in bioinformatics. So the first things that you need to assemble the sequence that come out, and after because you put lots of sample in only one sequencing run, you need to retrieve your sample inside the sequence. So there is called the multiplexing part. After that, you take off your sample, and there is a kind of some tag in front of the sample, can be the indexes. So you're looking for the index to retrieve, which index corresponds to which sample. So looking for this index give you the sequence that belongs to one sample. And after there is several filters that you can do during the bioinformatics. So you retrieve all the cameras, you remove all the sequence that can be PCR or sequencing errors. And after that, you clean all your samples. You compare your sequence from the sequencing with the reference database. And at the end, you have the list of the species that were present in your samples and the number of sequencing reads that correspond to those species. So you can have a kind of a semi-quantitative aspect of the number of the species that are present in your samples, and also on the percentage that are present in your sample. For example, species 1 is 20% of their sample, and species 2 is 80%, something like this. I don't know if it was very short, but I hope I answered the question.
Newton,
Newton,
ainda aqui no Brasil,
still here in Brazil, bioinformatics is a bottleneck. Not exactly because there's a lack of professionals, also because of lack of professionals, but the issue that frightens people the most, it's the computing equipment that is necessary for the analysis, because we have millions and millions of reads, right? And every read is a sequence that you have in your sequencing. So I showed a program, a very easy to use program that can be easily used by people who have basic knowledge of computer science. There are some limitations, of course, in this program, but taking this program and very few other programs around the world, they have a potential called genius. The others are relatively complex, and they require high capacity computers. I believe that in a few years' time, maybe 2 or 3 or 5 years, the situation is going to be much better compared to today, and definitely it was very bad 5 to 10 years ago. Yeah. Thank you very much. I remembered the question that I wanted to ask before, and I'm not going to miss my chance of asking it. When we collect eDNA in bio, I imagine that we can also collect the DNA of a species that is like 300 kilometers away in the beginning of the stream. Isn't this a little confusing in terms of our interpretations and our results? Dr. Alice?
Yes, there can be some drift of the signal of eDNA that's coming from the upstream to the downstream, but this can cause some false positive. However, the DNA is not all the DNA that coming from the upstream to downstream, just only from the species that very abundant on the upstreams, and some few molecules can come down in the downstream. So you need to when you collect, you should take this in mind, and there are some formula that allow you to calculate this distance of the eDNA signal, and this depends on the size of your river and the velocity of the river. For example, a small stream in a forest that's less than 1 meter wide, your DNA drift is very slow. This is not very big. In big river like the Rhone, we calculate that can be 100 kilometers, for example. But a recent study that we did in French Guiana again, we collect every 10 kilometers around the Maroni River and the Oyapock River. What we found that the DNA reef for the entire fish community is only a few 100 meters. There is some drift that come up, but the fish community signal is consistent with what it was founded with traditional methods. So this drift, you consider that the entire community is not a bias in your analysis.
A escala tem que ser outra, né?
Outro ponto.
It's a different scale, yes. And also repetition, right? If it is very important information for your work, You can repeat the test to see if you can identify it at that point or in other points, if you can see that DNA. And the scale cannot be that small, right? You have to consider a bigger area maybe, because when you have an animal, right? Yeah, Newton, that's exactly where the biological knowledge about the species comes in. So with the knowledge of the species, you're going to be able to determine a strategy. If it is a species that migrates a lot or very little, all of the data must be discussed with the specialists in terms of environmental DNA so that you can make up the best strategy to confirm your result. And maybe that's why these new techniques are always going to have to move together with classic techniques. Exactly, exactly. They don't compete, right? They complement each other. That's right. Okay, we have some other questions over here. I don't know how much time we have. Let me just check. Guys, how much time do we have? Yeah, we have time. Okay. Okay, so a very important question that is very different between Brazil and Europe. It's about the cost. And I've seen some work showing, Dr. Alice talked about it. The costs can be much smaller in terms of eDNA monitoring if we compare with the traditional techniques, but maybe in Brazil and with Professor Evanguedes, the reality, if we can actually have a smaller cost here in Brazil. I don't know if there is this difference, and what is the prognosis with regard to the cost of these technologies? Well, nowadays, we still have a relatively high cost because of importing of reagents. The taxes are still very high for importing the reagents. And there's another detail, the lack of specialized maintenance. So specialized maintenance of the equipment is concentrated, for example, in São Paulo. And if you have equipment and you need a technician, this costs a lot. So that's one of the points. In Europe, you have a larger number of centers, and Dr. Alice can talk about this much better in Europe. And this dilutes the cost for the company that produces and for the company that is providing the services. And another point is with regard to the number of samples. So if, for example, you analyze a single sample, depending on, well, we call it, for example, if you use a platform from Illumina for sequencing, depending on the cartridge where the sequencing is done, it can cost up to 12,000 reais in Brazil. And you put it in the machine, and there's no, if you use it once and that's it. You can't use it again. If you use one sample, the sample, for that cartridge, it's 10 to 12,000 reais, and the cartridge has the capacity of receiving a much bigger number of samples. So if you plan your work very well, how the collection is going to work, the marking of each point with the indexes that were mentioned by Dr. Alice, you can sequence, and you should sequence this at once, and then you can separate the different samples. And then your cost, even in Brazil, goes down quite a bit. And the expectation as well the expectation, just like in every technology used, the amounts, they go down over time. And Brazil has another problem, which is devaluation, depreciation of the currency, right? So everything is charged in dollars, and we have depreciation of the real that's very high. So this also increases, yeah. Sorry. Yeah, there is the possibility of sequencing abroad. There are specialized companies that do that, and you can also reduce the cost. The only problem is that you lose control of the quality and the way that you want the sequencing to be done, but it is a possibility. I think Dr. Alice can actually tell us about what happens in Europe.
In Europe, it's not much the consumable cost, but it's the personal cost that can be higher, I think, than Brazil. But comparing to traditional method, for example, for gray crested newts, the eDNA analysis was 10 times less expensive than a traditional method. Is that actually traditional method is the time of the person that spent it in the field. And this was for UK, but the price between UK and France are different. And so for the bullfrog was only 1.5 cheaper, 2.5, sorry, cheaper than the traditional method, probably because the survey method were different. So the time spent in the field was less. But yes, there are plenty of companies that propose the service of starting from the extraction to the bioinformatic analysis, and Spygen is one of those that, and people come to us to ask this because they found that still less expensive and more reliable than traditional method.
De uma forma geral,
in general, the whole process has to be evaluated, right? So you have specialized people. That's also a problem that must be well considered, right? And for you to maintain people who are specialized and well trained, you need to have a demand. And if you don't have demand, you're not able to maintain these people. So that's why the beginning of the process is always more difficult. But having a demand, you have, the cost goes down. Gabriela is asking, what should we do in Brazil for these costs to go down? So is it through a partnership with universities and spin-offs or the private sector, this sort of work? What is missing in Brazil for us to be able to offer and for the market to absorb these techniques, for this to be used in a more disseminated manner? I believe that university should work as a training center, as an educational center, but this work should be carried out, these activities should be carried out by a private initiative. I think it would be more efficient in this manner. I don't know what Dr. Alice thinks.
Yeah, I totally agree with that. We start Spygen as a spin-off of a new university, and now we work, we sell the service, but we still collaborate with university for the development of the method.
Muito obrigado. A gente tem mais uma
Thank you very much. We have one more question here from Vincent, and I think it's a great question. He asks about the precision and reliability of doing a quantitative analysis of the populations in the environment. So is this really something that is possible? It's a question for both of you. The quantitative determination of multicellular organisms is always more complicated because an individual can release more than one copy. So you never know if the number that we call REES, right, the number of sequences obtained, came from the same individual or from several individuals. We are working on this, but it really makes it very difficult for the methodology. We are placing more than one region in this analysis, more regions, more variable regions, for example, microsatellite regions, to distinguish between the number of individuals that have that sequence, because there's the expected number of variations, but it is really very complex. Now, for individuals, for microorganisms, unicellular organisms, it's very easy to be done. Dr. Alice, would you like to add anything to that?
Yes. Quantification is still a big debate, but more and more evidence found that there is a kind of a relationship between the biomass of the species present in the environment and the quantity of DNA detected by qPCR. But even with NGS, it was found a positive correlation between the biomass or the number of individuals and the quantity of reads detected. And we found that the DNA copy of DNA that was found in the Rhone River, for example, was really correlated with the biomass. And we found a similar trait in the Danube River. We did an experiment on the Danube River, and the quantity of DNA was correlated with the quantity of fish biomass detected for each species. But still, the topic is something like the holy grail of eDNA, and people start to work intensively on it in the last few years, so that's something that can be grow bigger and bigger in the next future.
Atualmente, a gente ainda depende dessa.
Currently, we still depend on the traditional techniques for these things. Yes. There's a question here in the chat. Let me just check here from Sylvia. And I thought it was very interesting because she says the following: maybe the residue from the trash, right, that is thrown away, does that affect the eDNA? For example, a river near my city. So, for example, they have fish in aquariums and they wash the aquarium, and, you know, how far is this a complication factor? You know, because this water goes down in the sewage, and how does that affect the detection of species? Newton, you can have a strong influence. If you imagine that even hormones that are used by women, they are detected in the rivers, and they end up modifying the sexual characteristics of animals. You know, you can imagine that there is some influence. So yes, it can happen. But normally in the case of the analysis of species, you would have previous knowledge of what the species, the cultivated species, are in that region. And You could even try to make an analysis of the wealth of DNA of these species that probably come from their breeding and collecting it in the source close to where they are raised, so you have an idea of contamination. I don't know if it's possible, but for example, if people, they eat fish, right? Salmon from far, right? Salmon that comes from Chile, or fish that comes from the ocean. In my city, we're in the middle of Brazil, and DNA fragments, maybe, you know, they come out in people's feces, right? So could this DNA travel, you know, all this?
Yeah. Yes, you can detect it. Yes, close to Lyon in France, there are plenty of marine fish, and Lyon is the middle of France, very far from sea. So you can find salmon, and you can find some bar or things like this, the red, every fish that people, tuna, yeah, tuna. So you can find it, but again, you know that you are in the middle of the fish that can be eaten by human are not so many, so you can found it quite easily in your sample and say, okay, this is coming from human consumption.
Ou seja, novamente.
So in other words, once again, it is information that professionals in the area should consider. So before saying, oh, so there is a marine species in the river in the middle of France or in the middle of Brazil, right? Yes. Yes, we think that tuna is from São Francisco, right? The São Francisco River. Yeah, that's also important for us to check, right? The importance that, you know, how important it is for people to work with this. It's very difficult for us to have this in the Brazilian market today because we don't have enough technicians that are going to interpret all of this, because it is very difficult. You know, there are several issues. For example, I saw a recent article where the authors say that the difficulty they had to identify felines. So I don't really remember why. I don't know if you know about this article, because there are some groups of animals that are less identified compared to others. So that's also a difficulty. Dr. Alice actually said something about the difficulty in the beginning. If you don't have a database, a specific database about the species or rare species, or even if the markers don't have a differentiation of the species, you know, you're going to be in doubt about what species you're working with. Yeah, because I think that, for example, in the Amazon forest, you know, 3 fingers below the ground, you know, it should spread, you know, less DNA compared to a plant, for example. Yeah, it really depends on the person knowing the ecology and the biology of the environment to be able to interpret all the data, right? Exactly. We have a question here I could have asked before. It's about filtering, automatic filtering. And Isabella is asking Dr. Alice, do you have any comment about Strivex compared to the automatic filtering system? I don't know what Strivex is, but probably you know.
It's a filter, and I don't know what is automatic filtering. Maybe it's the filtering that I presented in the presentation. So with Strivex, you can only filter 2 liters of waters. With the filter I presented, you can go up to more or less, we do 30 liters of waters, but when water is completely clear, it's probably you can go up to 60 or even 100 liters of water. So the difference is the quantity of water that you can to pass through the filters. So it was a part of my presentation when I presented the two marine study. The first marine study, we used Sterivex for collecting water sample because we didn't have at that moment the same filters that for the river. For marine environment with 0.2. So we developed later on. And the second project, we used the 30 liter of water filters. And so what we found that we collect more or less the same, the volume of water that we collect is more or less similar to have the description of the biodiversity of fish. But with Sterivex, you need much more Sterivex to achieve this quantity. Instead of the 30 liter water filters, you need less filters. That's more or less the difference between the two, but to work quite well.
A bomba que você usa é uma bomba.
So the pump that you use is an aquarium pump, sort of?
Not really. It's a kind of peristaltic pump that's make void, and so it brings the water with the void inside the filter. So there are plenty of model of this. The boat is one of them. We have this drill. I don't have any picture with me, but it can be used. But there is also the picture about the Brazilian frog, where the person was doing the sampling. We was using again another peristaltic pump. So there's something like that clamps the tube, and clamps in the tube is making the void and suck the water inside the filter.
Certo.
All right.
Eu gostaria de fazer.
Yeah, I'd like to ask Dr. Alice a question. What kind of solution do you have or have you used for water that is very dirty, as in the case of several rivers and lakes in Brazil?
Actually, we have a DNA extraction protocol that's get rid of the inhibitors that were present in the DNA coming from those kind of samples, but we don't have any particular solution. Actually, we use the same solution from marine water that are crystal clear and from Guyana River that I think is much similar to the Brazilian one.
E sobre o entupimento do filtro?
What about when the filter gets clogged? The particles, they can, in a few mLs, you know, depending on the place where you work, they can clog the filters.
That's why the filters are so big, because the membrane is 500 square centimeters. So the clogging starts after several liters. So it's not get clogged so fast. So that's why there is big and several times folded. So there is not like the flat filters that can be clogged after a few meals.
Obrigado.
Thank you.
Tem uma pergunta da Sheila que eu acho.
There's a question from Sheila over here. Her question is, she says, I work with vertebrates, and is it possible to collect environmental DNA of the sediments? I think so, right?
Yes, it's possible to collect DNA from sediments. However, you should consider that in water, the DNA can degrade in few days or few weeks, but in sediment can stay very longer, in perfect condition can say 100,000 years. So it really depends on the condition of the environment. And in sediment, you are not sure that what you are collecting is the real community or the past community. So, but really depends on the condition. As Dr. Evanguedes, sorry for your name, said before, when it's wet and warm, the DNA degrades faster. In dry condition and cold, DNA stay longer.
Obrigado.
Thank you.
Tem uma outra pergunta também muito interessante.
There's another very interesting question here, which is about fungus. So Enyo is asking about, are there studies that use the eDNA technology for fungus? I had a professor in the university, and that's one of the few references we have in Brazil in taxonomic studies of fungus. So do you think the use of this biotechnology can help with taxonomy? Because we definitely don't have a lot of that in this area. I thought it was interesting. For taxonomic reviews, it's for both of you. And in fact, the profession of a taxonomist, I've seen a lot of work showing that it is going down, right? There are less people who want to work with taxonomy. And, you know, to what extent can a taxonomist use eDNA to identify species in the environment? There is something I call initial work. Very few people like to do that, which is to extract the DNA of a species, morphologically characterized or biochemically characterized. You have to describe the DNA of the species. You have to place the results in databases, and then eDNA can identify that fungus or any other species. So the taxonomist would help a whole lot at this point, if he could also accept the idea and if he could work in collaboration, providing samples that are well characterized for those who do the sequencing so that you can generate data and you can place this data in the database. Dr. Alice, would you like to add anything?
No, I completely agree.
Deixa eu procurar mais perguntas.
Let me just look up others. I only have one question about fungus. I don't know if Dr. Alice has worked with fungus, but if you work with molecular biology of fungus, they suffer a lot because the purification of DNA from a fungus, it's always a little more complicated. It's always a pain because you have to break down the DNA protection. For the fungus, it's much more difficult compared to a mammal cell or even a bacterial cell. So this is one of the issues, and I don't know the consequence with regard to eDNA studies with fungus. So the DNA of fungus is always more protected compared to other species or other groups. So false negatives would be more common, maybe. Well, I think so. I think we should have a specialist about fungus to confirm this here. I don't know if Dr. Alice has any idea about this.
We work very little on fungus, but we usually work on extracellular DNA from fungus. So we said that we think that nature already done it, the tough work about the extracted DNA from the fungus. So it's much easier.
Claro.
Yes, of course. Okay, I think our time is almost up. I'd like to know if you would like to make any final remarks or any final issues you'd like to talk about. I have a comment, one or two actually. The first one is about the importance of this sort of meeting, not only in Brazil, but for the whole world. It is a technology that definitely has several applications. It must be used more. It must develop social, environmental, and economic development because it has this capacity. So in terms of the use of this technology, it's very important to have meetings like this, and this is all very positive to clear up these questions we have. And fortunately, this is going to be recorded, and people who are curious about it, they will be able to watch us and learn a little bit more. And I would like to ask Dr. Alice to, in some way, leave us a little bit more about the company that she works in, because it could be very useful, not only for our group, but also for several other groups.
So your question is knowing more about Spygen. This was the question.
Isso. Esse foi o comentário.
Yes, exactly. That was the comment.
So Spygen is a mission-led company. Actually, it's kind of hybrid between a real company that sells the service, but the income that we have are used for our mission. Our mission that we have is to use eDNA to better knowledge about the biodiversity. So we use the income to fund scientific project or scientific expedition around the globe to use environmental DNA to describe the biodiversity, and to also participate to scientific publication. And so we were founded in 2011, and by two persons, and I am one of these two. And now we are something like 60 people working on it. And so we sell the service mostly in France, but also abroad, mostly in Europe. I don't know if this answers your question.
Responde sim.
Yes, it does. And you have already talked a little bit more about the company, about the idea of how the company works. And if possible, it would be interesting for you to leave your contact for those who are interested to write to you and maybe request help, or maybe to propose a collaboration.
Yes, of course. So if people are interested in what we are doing, just write me an email. It's quite easy. It's alice.valentini@spygen.com. And so I will try to answer your question, or if I'm not able, I will forward to the person that can better answer you. So, but it's just my name with dots and the company name is my email address.
Obrigado.
Spygen.com.
Thank you. Yeah. Spygen.com. I'm going to put it in the chat here.
Okay, yeah. Yes, that's the name.
Exatamente, é esse. So, Professor Evanguedes, people are asking for your contact as well, if you can leave yours. Oh, yes, mine is my surname at gmail.com. So I'm going to write it in the chat here. All small letters. It's my surname at gmail.com. Okay, thank you very much, guys. And I was thinking about how important it is to disseminate this idea. And what came to mind was the importance that it has in terms of spillover areas where we have, epidemics and pandemics nowadays. So that would be a more dynamic way of monitoring these areas, right? Well, anyway, it's almost 200 people online here. I think it's a very good number, very participating people who participated. There are more questions, but we don't have a lot of time. So I would like to thank Professor Evanguedes and Dr. Alice for your time, for being here with us and answering questions. And the Center of Bioengineering, thanks you very much for your time. And we're going to see each other at other events. I hope so. I hope so. Thank you very much. Thank you, Dr.
Thank
Gabriela.
you very much. Thank you very much for all the organizers.
Bom dia.
Very nice event.
Gabriela.
Gabriela, over to you. Okay, guys, so we have finished our first day of the event. Thank you once again, Dr. Evanguedes and Dr. Alice Valentini for their participation in our event. We know how much time is important. Thank you for joining us, and we'll be back tomorrow on Tuesday at 10 a.m. with the presentations in the roundtable. Tomorrow we will have Dr. Guilherme Oliveira from the Institute of Technology Geo Valley and Ryan Silva de Paula, our researcher. We also have presentations and a roundtable, the same format as today. Thank you all for joining and hope to see you tomorrow. Have a great day.